Thank you, Michael, and thank you very much to the organising committee of the Alliance for Regenerative Medicine for giving Cynata the opportunity to present to you today. Those of you who have followed our journey will realise that we remain the leading company worldwide in the development of iPSC-derived cell therapies, utilising a platform that we call Cymerus to manufacture and ultimately use in clinical applications an iPSC-derived mesenchymal stem cell product. Before I get underway, please do refer to our forward-looking statements, our website and the Australian Stock Exchange platform. A few words about the company to provide an introduction. We are listed on the Australian Stock Exchange. We IPO'd back in 2013 and our market cap is around $27 million US. As I mentioned, our technology platform, which we call Cymerus, utilises iPSCs to manufacture a highly consistent and efficient allogeneic MSC product. The technology originated at the University of Wisconsin in Madison, the home of stem cells, even though we are an Australian company. We have developed a compelling preclinical data package in proof of concept studies and followed that with a world-first clinical trial in acute graft versus host disease. We have a strategic manufacturing partnership with Fujifilm and a robust clinical pipeline with products in phase 1 through to phase 3. That manufacturing platform, as you would understand, utilises a single iPSC cell line derived from a single donor from a single donation. The strong efficacy and safety databases allowed us to move into multiple clinical trials, including a phase 3 in osteoarthritis. The market opportunity of those significant unmet medical needs is very attractive, around $25 billion US, and significant value upside for our shareholders through our business strategy of strategic partnerships. The company recently raised some additional cash and we're well placed to deliver on milestones that I'll talk more about shortly. Evie gave a nice summary about MSCs and their mechanism of action. And for the empiric pharmacologist there, we've got to think beyond just a ligand-receptor interaction to explain mechanism of action here. There is now an enormous body of literature which confirms the immunomodulatory properties of MSCs, particularly through elicitation of immunomodulating cytokines, phenotype switching of effector cells, and many other actions, which again have been confirmed in the literature. This is, of course, a result of the extensive interest in the clinical potential of MSCs. We now have around 1,200 clinical trials having been registered with MSCs in the past decade, and products now on the market in Japan and Europe. But where the challenges arise for cell therapies generally, and MSCs in particular, is the manufacturing process. In our view, and as I'll also explain shortly in the view of the FDA, the conventional process, which relies on donated tissue such as bone marrow or fat tissue, is suboptimal and flawed in many ways, primarily because of the low numbers of MSCs which can be derived from a single tissue donation and the fact that multiple donors are required, which leads to batch-to-batch inconsistencies. So the manufacturing challenges require a different approach. And this is where Cynata's technology platform comes to the forefront. On this slide, you'll see in the top panel a schematic of the conventional process that is used today by companies that rely on MSCs from tissue sources. Donors are screened, tissue is derived, the MSCs are purified using one form or another, and then expanded extensively in culture to produce sufficient quantities of cells, bearing in mind that a bone marrow donation will typically yield only around 20,000 MSCs, and a typical dose requires many orders of magnitude above that. So those cells have to be expanded in culture, and that's a flaw in and of itself, because MSCs, whilst they do have some very important properties, including an ability to replicate, that ability is limited by the senescence and the reduction of potency after only a few rounds of cell replication in culture. Moreover, a reliance on multiple different donors has an obvious flaw. Every donor is different, and therefore every MSC derived from different donors have slightly different properties. And a cardinal requirement, as many of you will be aware in the pharmaceutical industry, is an absolute requirement for manufacturing consistency, and that cannot be assured utilising a strategy that relies on multiple donors. In the lower panel, and conversely to the MSC derived from bone marrow situation, Cynata's utilisation of iPSCs as a starting material ensures we have batch-to-batch consistency because we're always using the same starting material, and the expansion of the cells is typically occurs at the iPSC level. So we can expand iPSCs without alteration, and then the last step in the patented process is to differentiate those iPSCs into MSCs, thereby ensuring we have a highly consistent finished product and a highly potent finished product. I mentioned the FDA's commentary around the manufacturing strategies that rely on donor-derived tissues, and it was very clear from their commentary a couple of years ago in relation to a BLA application that had been submitted to the FDA that, as the FDA put it, the functional heterogeneity that arises from donor-derived strategies is flawed. Unless the manufacturer can demonstrate batch-to-batch consistency, which fundamentally is impossible given the differences between donors. From a cell therapy development point of view, this sort of commentary from regulators is extremely useful because it gives us a window through, in this case, to the FDA's thinking around what we and other manufacturers will need to demonstrate when we come to seeking market authorisation through the BLA process. So obviously that's been very helpful for Cynata and the field more generally. Looking now at how we've been able to move into the clinic with our iPSC-derived MSCs, we have 4 clinical programs underway at the moment, 2 trials actively recruiting, 2 more that will get underway this year. And then we have 3 additional clinical targets that we have a substantial preclinical database in support, which we will develop into clinical programs based on our partnering activities. I'll talk more about a few of these indications now in detail. Our first clinical study and a groundbreaking clinical trial was in acute graft versus host disease, patients who had failed standard of care at that time, which is corticosteroids, so a very, very grim outlook for these patients. We achieved all of our safety and efficacy endpoints at day 100 and in follow-up at 2 years, a complete response in these patients, 53%, so more than half of the patients had complete resolution of their symptoms, an overall survival, an overall response rate in excess of 80%. When we looked at those patients after 2 years, it was very gratifying to see nearly 2/3 of them were still alive, compared with standard of care where less than 20% are expected to survive out to 2 years. That also compares, our result at 60% compares very favourably as well with the reported data from other MSC products, which is less than 50%. The fact that this was the first completed iPSC-derived cell therapy clinical study caught the attention of the editors at Nature Medicine magazine, and as you can see on the slide there, we had a front cover article back in 2020. We'll be following that up with publication of our 2-year data as well. That substantial result has allowed us to move forward now to a planned phase 2 clinical trial in acute graft versus host disease following clearance of an IND in the US last year. We expect to get that trial underway in 60 patients in the next few months at sites across Europe, the United States and Australia. We also have underway a clinical study in diabetic foot ulcers. As we heard from Evie, this is a significant unmet medical need wherein the standard of care really hasn't changed in the last 100 years. It's dominated by dressings and application of bandage-type device products which really haven't resolved this very, very challenging medical condition. This study is being conducted in Australia in 30 patients who have been randomised to receive either our product, which is a topical application of our MSCs in a proprietary patch, or standard of care over a 4-week period. And I'm pleased to say that we now have some initial clinical data from the first 6 patients, 3 of whom received our product and the other 3 received standard of care. And as you can see here in this clinical study, the top panel shows a patient that had received our product, CYP006TK, at day 0, classic sign of a non-healing diabetic foot ulcer, and on the right-hand panel at day 28, the wound has substantially healed. Bear in mind, these patients are enrolled only after they have failed standard of care for at least 1 month prior to enrolment, so these wounds normally don't recover particularly well. In the lower panel, you'll see the patient with standard of care, and that wound really hasn't advanced very much at all. Presented graphically across the 6 patients, bear in mind this is early data, so it's only in 6. We have a total of 30 coming up. When we complete the clinical study, you can see that at all time points, the mean surface area of the ulcer has improved significantly, or I should say substantially, over time compared with the placebo or standard of care group in the lower bar there, the light blue colour. So obviously we're guarded about just 6 patients, but it's still very encouraging to see this sort of response in the patients so far. And clearly, if this is carried through to the full 30-patient data set, which we expect later this year, then this will be a very, very good outcome for this clinical trial. We have a phase 3 clinical study underway in Australia in osteoarthritis. Again, as we heard from Evie earlier, osteoarthritis is managed primarily by pain relief, but it does nothing to the eventual outcome in these patients whose quality of life is significantly diminished by the degenerative nature of this disease. If a cell therapy can result in a disease-modifying outcome, then this will be a substantial breakthrough in the treatment of these patients. We expect to enrol the full complement of patients for around 440 toward the end of this year, and following readout towards the end of 2025, early 2026. The 4th clinical program we expect to start later this year is in a group of patients who have had a renal transplant. As you would know, organ transplantation is associated with lifelong anti-rejection therapy, and the goal here is to ameliorate that requirement with the use of MSCs. That will be in 10 patients in Leiden University Medical Centre in the Netherlands. We're often asked, well, how do you know the cells that are manufactured utilising the Cynata process are better? Well, ultimately that requires a head-to-head clinical study, but we do have preclinical data which supports the thesis that the cells are more potent. Here in this study, in looking at left ventricular function in an animal model of myocardial infarction, where we saw a significantly greater response in the animals who had received our MSCs versus conventionally derived bone marrow MSCs. Similarly, in a wound care model, very pertinent to our diabetic foot ulcer study, where our Cymerus MSCs showed a much higher rate of wound healing compared with bone marrow-derived MSCs. For comparison, we looked at a couple of other sources as well, dental pulp, gingival fibroblasts, and bone chips, which had greater or lesser potency in this model than did the MSCs, but they don't represent viable commercial sources. We have a strategic manufacturing relationship with Fujifilm. As we've heard in the course of this conference over the last couple of days, manufacturing of cell therapy products is key to their ultimate commercial success, and Fujifilm is growing as a powerhouse CDMO in this area. Looking ahead at near-term catalysts for the company, completing our recruitment in our diabetic foot ulcer trial, and then ultimately reporting those results, commencing our phase 2 clinical study in acute GVHD, completing recruitment in the osteoarthritis study, and then ultimately moving forward into the renal transplant trial with Leiden University. Quick corporate overview, we have 3 major shareholders, Fidelity International, one of the largest institutional funds worldwide, Bioscience Managers are a specialist fund in Australia and the UK, and Fujifilm, our strategic manufacturing partner. I'd like to conclude there and look forward to following up with any of you who have any questions or comments during the course of this morning and later today. Thanks again for your time.